Good morning, brain-in-laws. Welcome to day 5 on Planet Brain, OHBM 2021. What a week it's been in this crazy space-time continuum we've been in. Couple of reminders. All posted presenters today are invited, if they wish, to stand by their posters during the poster sessions, regardless of the category. To bring in more interaction and chat if they wish. I would like to also show a couple of slides just to re-emphasize who was behind this and who made this possible. And first of all, of course, our sponsors and our exhibitors who we have a symbiotic relationship with, and we appreciate them very much. And I urge you to go and look at some of their wares. I've been in there and I've been downloading stuff, and I've already made contacts for any Zooms next week. Check it out. We have a competition as well. If you haven't remembered. You can win an individual or group registration to our Glasgow 2022 meeting. Visit the exhibitors in the exhibit hall and get a badge from them. Okay, and now on to our next slide. This is a huge thank you to all of the volunteers who worked on this meeting, the program committee, the OHBM Technology Task Force, and program committee was headed by Daniel Margillies. The tech task force was headed by Alex Fornita, and they were basically worked as one across time zones for since August 2020. Daniel was also an integral part of the technology task force and part of the OHBM by Sparkle Special Ops unit that we had, because this was a very tricky enterprise. Sparkle and our OHBMX volunteers had to work together for coding to happen, and we had a few hurdles, but we managed to get a really good thing going this week. So I really thank everybody for that effort. And of course, that also includes the huge load that was carried by our SIGs, our Open Science SIG, Student Postdoc SIG, the Brain Art SIG, and our new shiny new Sustainability and Environment Action SIG. A big thank you to everybody. It takes a village, it takes a planet. Now, I will introduce Christoph Phillips, who will introduce Nikolaus Weiskopf. Christoph.
Hello, everyone. Welcome to this keynote presentation. And to me, it's really an honor and a great pleasure to welcome Nikolaus Weiskopf. I think this is also the second presentation of Nick's keynote lecture. And so it's for those who haven't had the chance to listen to the first one, this is your second chance. In order to introduce Nikolaus, I have the chance to know Nikolaus quite a bit. I collaborated with him, still collaborating with him. But for those not in the know, so Nick is currently the director of the neurophysics department at the Max Planck Institute for Human Cognitive and Brain Science in Leipzig, Germany. He's an expert in obviously magnetic resonance imaging methods and the applications to neuroimaging. And what is really outstanding is that together with his collaborators, he pioneered a few research tracks in MRI, such as neurofeedback, and he's really able to make the bridge between basic MR physics and potentially clinical applications. So his current research focus is really to develop functional anatomical microstructure imaging using MRI. The point is to really study the structure, function in the brain, and the interplay with the microstructure level. He overall, over the years, well, he obviously received this PhD from the Max Planck Research School in Tübingen. He did a few postdocs, and then he ended up with a faculty position at the Wellcome Trust Center for Neuroimaging at the Institute of Neurology, University College London, UK. That's actually where I met him. And then he was appointed as director at the Max Planck Institute in Leipzig in 2015. But he's kept good links with universities he worked at. So he's the honorary professor at the University of Leipzig, the University College London, adjunct professor also at the Medical University of Vienna, member of Young Academy of Europe. So I would say that he's a star in his field. I want to also say something about his ability at mentoring. I mean, beyond is great and really impressive scientific output. Nick has been a really good mentor to many researchers, and some of his mentees have landed top positions in various labs around the world. So today, his presentation really will focus on understanding the normal and diseased human brain, and it's going deeper than the image, really, the topic. So looking at the microstructure in the brain using in vivo technology, while most of what has been done in terms of microstructure has been more focused on ex vivo histology. So he will discuss how this interdisciplinary approach and how to develop novel magnetic resonance imaging acquisition methods, the image processing that goes along, and the potentially integrated biophysical models to establish this non-invasive quantification of histological measures of brain tissues. And without further ado, welcome Nick, and I guess he will give a short or a good summary, a wrap-up of his pre-recorded presentation.
Chris, thanks a lot for the very kind introduction. I hope you can hear and see me. I will share the slides now with you.
We can hear you, Nick.
Excellent, great. Yes, indeed, I'm going to give a wrap-up or a live talk summary of the pre-recorded presentation, which of course contains much more detail, which I can't go into here. So please feel free to go to the pre-recorded talk as well for for some of the further details. So thanks a lot for the kind introduction, and I would like to pick up one point you made about mentoring and you know, kind of training the next generation of scientists. And I wanted to say that actually for me, OHBM was one of the central conferences that really shaped me as a scientist. And I've really grown up with it. And I just had a look at kind of my OHBM attendances, and I realized it's now 20 years ago I attended the first HBM, which was in Brighton at the time in 2001. So I hope I can give a little bit back of that now, what I benefited and learned from HBM. And I'm very honored and very pleased to have the opportunity to talk about our work on microstructure imaging here. So let me, oops, let's see. These are my disclosures. And what I'd like to do is give you a short primer into why we are interested in
Nick,
in
we
vivo
can't see your
histology.
slides.
Oh, you can't?
Sorry, no.
Okay, sorry. I'll try that again. Does it work now?
Yes, we
Yes.
have your slides.
Okay, perfect, great. Thanks for letting me know, Chris. So these are my disclosures, and I'd like to give you a little primer on why we are interested in microstructure imaging, although Chris already said quite a bit about it, why this is such an important topic for us, I think, as a group, but also as a community, obviously. And then I'm going to give you a few examples where we developed techniques for that, how we applied them, and wrap up with a discussion at the end. And so why are we interested in microstructure imaging? Well, when you want to understand the brain, basically you have this issue that the brain is a highly complex organ where you have to cover about 6 orders of magnitude of spatial scale to really understand it, going from the macro scale, from brain areas, down to the micro scale, where you look at cells, where you look at axons, for example, and fibers. And MRI is doing a great job, especially high field MRI, pushing into this mesoscopic scale where we can start looking at, for example, layers, kind of intracortical features. This may also include, for example, columnar features like here the ocular dominance columns. But what we really need as a neuroscientist is also this access to the microstructure itself. And here, obviously, we are not going to achieve voxel resolutions that resolve, at 1 micrometer resolution that would directly resolve these structures. But what we can do, and what is amazing about MRI, is that we can still get from the rich data we have in the MRI, from the MRI signal, we can estimate using the right biophysical models some aggregate measures of the underlying microstructure. So we can kind of indirectly see that. And why is that the case? Well, in a nutshell, and I explained that more in the pre-recorded talk, but in a nutshell, we really benefit from the fact that MRI is sensitive to a lot of different physical interactions. So what does that mean? Well, we basically record the signal from the spin of the proton, and typically the proton in H2O, in water. But water lives in different environments. It can be, for example, close to myelin sheets, where you have a lot of macromolecules, where water is restricted in its mobility. Or we can have, for example, myelin sheets that also prevent water from freely diffusing. And what we can do with MRI, we can sensitize it to these different physical interactions. So that's special about MRI. Even though we measure the signal from the same spin system, it depends on the environment, and we become sensitive to the environment. And for example, magnetic inclusions like iron also impact the signal. And you can visualize that, for example, and create and sensitize MRI to these specific aspects. For example, here the longitudinal relaxation rate or the magnetization transfer saturation that is dependent on the mobility, for example, of water. Then the diffusion imaging, quite well known, is dependent, for example, as you can see nicely here in the corpus callosum, depends on what type of diffusion barriers you have, in what direction they point, or here you see the example of, for example, the apparent transverse relaxation rate being sensitive to the higher ion concentration in the basal ganglia. So now we basically have a non-invasive tool to look at microstructure from different points of view. That's how you could think about MRI. And that's a real strength. And how can we use that? Well, we can try to solve this really difficult, notoriously difficult problem of going from MRI data to underlying microstructure. And this is, you can view this effectively as an inversion problem. So if you knew exactly the underlying microstructure, we would calculate the typical MRI parameters I presented to you before, and from that, the weighted images. So kind of the quantitative images and then the weighted images. And then you could regard going from the weighted images to the underlying microstructure as an inverse problem. And that's hard to solve, it's ill-posed, but you can kind of condition it better by acquiring the data with different types of contrast. So it becomes a better posed problem in the end. And in addition to estimating the microstructure, what we are very keen on is understanding structure-function relationships. So we are also working on models that connect it up to function. And this could be relatively simple models, but this can go all the way up to formal, like neural mass models, for example. So this is really what we are trying to do. And there's, of course, a lot of MR physics, biophysical modeling, and neuroscience proof of concept studies involved in that. And I will show you some examples of that. So let me talk a little bit about cortical myelin mapping now. So going to very high resolutions of 400–500 micrometer isotropic resolution at 7 Tesla, using optical perspective motion correction to avoid motion artifacts, you get these kind of maps, quantitative maps here. And I hope what you can appreciate from it is already going here from kind of the vertex to the caudal part of the brain. You can see already substructures and features within the cortex, quite a rich substructure in the white matter, for example. And when you go here and you look at the basic ganglia, you can really see them pop out, also substantia nigra here, because of the high iron content. So you can see these different viewpoints onto the underlying microstructure at a mesoscopic resolution. And you can exploit that, for example, by extracting the R1 values as a metric of myelination, project that onto the cortical surface, as you can see it here on an inflated brain. And you can see that the bright areas are the ones with high R1 values that would be indicative of high myelination. And you can see how, for example, the primary sensory areas pop out, the motor cortex pops out, and also primary visual cortex here, which with a highly myelinated line of Chinari pops out. And these data have been recorded in collaboration with Marty Sirino and Fred Dick in London at the time at 3 Tesla. So this is 3 Tesla data. What you can do here, first of all, you can see that it's well aligned with what we know from postmortem histology, but you have the advantage that you can obviously, because it's a non-invasive technique, you can run additional experiments on your subjects. And here we did fMRI, so-called retinotopy experiments, where we defined functionally and completely independent of the anatomical data, the primary visual cortex. And when you take the delineations of V1, project them back onto the R1 myelin maps, you can see how well they are aligned. And this gave us confidence in these type of maps to be used as myelin maps. And in addition, for us, it was one of the first experiments where we said, okay, we are really starting looking at now microstructure function relationships here. And of course, you can now dig deeper and say, well, if this is a myelin marker in one way or another, maybe we can learn other things from that as well. And that's what Saskia Helpling set out at the time. And she asked this question, is there a relationship between the myelination level measured with MRI non-invasively and the response amplitudes in MEG, here in particular in the auditory cortex? And she looked at several subjects and hemispheres, and indeed she found a correlation between the two. And we speculated, or we hypothesized to see this correlation, as I explained in the pre-recorded talk in detail, but fundamentally it comes down to relationships between pyramidal cell density, myelin density, and the response amplitudes driven by the pyramidal cells in the MEG. So now we have learned that we can actually use and learn something about the viability, about the features itself, and exploit that to look at structure-function relationships. Further encouraged by that and the fact, oops, okay, it's forwarding, great. So further encouraged by that, we asked the question, now we can look at entire brain areas, we can look at their features, but can we also look into brain areas and look at substructures there? And one of the substructures that comes to mind there is the so-called stripes in the secondary visual cortex. They are special in a sense, as we know from animal experiments, that they subserve particular functions, and they also show different anatomical features and microstructural features. But in humans, that's not completely known yet, and there's quite a bit of a discussion going on this. So Daniel Hanold in our department set out and went to used our 70 scanner to define the stripes based on function experiments, for example, color selective thin stripes, the disparative selective thick stripes, which interdigitate with the third type of stripes, the so-called pale stripes, and ask the question, do they show, for example, different myelination levels? And so he used these definitions based on the functional scans, defined ROIs, and then measured the R1 values in there. And as you can see, they're indeed lower for the thin and thick stripes compared to the rest, so to the pale stripes there. So being indicative of a lower myelination in R1 there. And Daniel's presenting that here at the conference if you want to learn more about it. So this is kind of should exemplify to you that you can even kind of zoom in and look at substructures in an area that are only about the size of a couple of millimeters or so. So we're really getting there now. And another example I'd like to present with these kind of techniques, and one advantage is that they're pretty versatile. You can apply them to different patient populations, healthy subjects, post-mortem imaging, but also even in different species. And this is kind of a special large-scale study we are running here with our colleagues of the Max Planck Institute of Evolutionary Anthropology, where we look at brains post-mortem of wild chimpanzees and after they've died of natural death. And this is special in a sense that our colleagues, they actually monitor these cohorts of wild chimpanzees very closely. So they know a lot about their life experience, for example, their experience with using tools, with social interactions, for example. So you can run studies now on looking at basically ontology, phylogeny, comparisons against other species like humans, but also take these special factors into account. And I just want to show some of very hot off the press data we have here, where you can see, for example, an aging study now in this cohort of chimpanzees. And you can see, for example, R2 star increases that would be in line with an increase of iron concentration in these different brain areas with age. And this looks very similar to actually what we observe in humans. And it's interesting to see that we also observe that in chimpanzees. And we also the differences between the different brain areas. Again, if you're interested in more details, please look at the posters by Ilona Lipp and Cornelius Eichner, who looks at, for example, connectivity in those brains and performs spectrography. And of course, as another aspect, what you can do is you can do in-depth histology in those brains. And here's just a nice example where you can see, for example, the line of Chinari in V1, which disappears in the secondary visual cortex, which is also reflected here on these R2 star maps very nicely. So I'd like to wrap up this part of the presentation briefly. So with the quantitative MRI approach, you have this advantage that you have more standardized data that is better comparable. And again, look at the pre-recorded talk for more details there. I hope I could convince you that we really have complementary views on the underlying microstructure. And I presented quite a few examples of R1 maps being used as myelin markers in the cortex. This is not a trivial one-to-one relationship, but it's quite a sensitive measure for it. But we can also look at now microstructure function relationships because we have a non-invasive marker for microstructure now. And it's a rather versatile technique. And again, if you're interested in other patient populations, diseases using these multi-parameter mapping techniques, please have a look at these abstracts as well at HBM. Now I'd like to kind of dive a little bit deeper into the brain, just underneath the cortex, where you have the so-called superficial white matter, which lies between the deep white matter and the cortex. And it contains the U fibers that connect adjacent gyri. There are very important connections, just the number of them is much higher than what you find, for example, for the long-range fibers, which we usually would track with diffusion tractography. They are important for the cortico-cortical connections. They're late myelinating. And they're affected differentially for different types of neurodegenerative diseases. So it's important to study them, but they're relatively understudied. And the reason for that is because we don't have so far, or just until recently, we didn't have reliable in vivo techniques to study them. And why is that the case? Because we have complex MR contrast mechanisms there we need to fully understand. When you look at diffusion-weighted imaging tractography, we have a crossing fiber situation that is difficult to resolve. And it's a very thin structure of about 500 micrometer extent, which you can only really resolve with very high spatial resolution. So these are all pretty big challenges for MRI. Evgenia Kirillina in our department, she was not discouraged by that and nevertheless tried to resolve these structures. And she observed one phenomenon also other groups have observed is that when you look at susceptibility weighted images or R2 star maps, QSM maps, you can see just underneath the cortex this hyperintense stripe. The cortex is highlighted in red. And because the susceptibility values are increased, this is indicative of that iron levels should be increased in this area. And the area is exactly where you would expect the superficial white matter. And she went into detailed investigations of using post-mortem MRI, histology, advanced histology methods, quantitative histology methods. You can find all that in the pre-recorded talk. But in a nutshell, what she found out is about this special contrast, that it's a particular contrast for the superficial white matter, and it's related, the R2 star value, to the fiber densities and to the maturity of the fibers, now speaking in a simple way. And with that knowledge, you can go in armed and perform now in vivo experiments. And what she exactly did. And here you can see, for example, a projection onto the cortical surface of the R2 star values in the superficial white matter. So it's kind of our superficial white matter marker here now. Here for comparison, it's the intracortical R1, our myelin marker intracortically. And what is quite interesting is that, for example, in the association areas, you find high R2 star values in the superficial white matter, which is compatible with U fiber rich association areas. But for example, in the primary visual cortex, you find very little. Very low R2 star values. And that's exactly what we would expect, because we know that there are fewer U fibers in the primary visual cortex than in the other areas. So it really fits the histological data as well. So this tells you something about kind of the features of and the densities of the superficial white matter. But what about the connectivity? To study the connectivity of those U fibers in superficial white matter, you have to go to diffusion imaging, and you have to really bring out the big guns here. And because of the high resolution, you need the crossing fiber issues. And we used one of those 300 millitesla per meter connectome MRI scanners that allow you to acquire diffusion data with very high quality, as you can see here, compared to, for example, standard diffusion imaging, because you can shorten the echo time a lot. And we combined that with a flexible RF surface coil, which fits around the head precisely and increases the SNR in the occipital cortex in particular quite a lot. And with both together, you can actually trade the higher SNR and quality off for resolution. We could acquire 800 micrometer resolution diffusion data and perform tractography here. This was done by Farah Movahedian Attar in our department. And you can see these beautiful images of actual U fibers, how you would expect it from histology. And that in itself, I think we were already fascinated by. But now, by now you probably know the game already. Of course, the next step was, does that tell us something about function? And indeed it does. And to learn about that, we performed additionally at 7 Tesla, again, an fMRI experiment, so-called retinotopy, as I presented before, which tells you something about what part of the visual field of view is projected onto what part of the cortex, where is it processed? So you can have kind of this relatively ordered relationship between outer visual field of view you look at and the cortical representation. Thus it's called retinotopy. And you find this retinotopy not only in the primary visual cortex, but also here in the secondary visual cortex. And the question we asked was, how are the two connected via the U fibers? Do we preferentially find U fiber connections between, let's say, the same visual hemifield of kind of visual segment between V1 and V2, or between different segments? So in other words, does segment 1 of V1 connect to segment 1 of V2, or does it connect maybe to segment 3? And this can be expressed into a connectivity matrix. And we hypothesized that you would mainly find the diagonal elements, so that you have mainly retinotopy connections. And indeed, that's what we found pretty reliably, because it was independent of the actual imaging setup. Different RF coils were used, and the experiments were reproduced. And even the experiment was reproduced in a completely independent sample of subjects. So this shows you now that you can actually really learn something about structure-function relationships and connectivity relationships between function and structure using these techniques. But you really need to push the resolution. And now we have, in our opinion, really interesting tools at our disposal to study U fibers non-invasively in vivo and learn more about their functional role. The last example I'd like to present is on now really going even deeper anatomically into the brain, into the midbrain structure, the so-called substantia nigra. And also in terms of the resolution, we are now digging even deeper in, and we're trying to get information about cells actually and cellular contrast. So what are we talking about? We are talking about the substantia nigra. As I said, it's a midbrain structure, plays an important role in the cortical basal ganglia thalamic motor loops. So it's, for example, also involved in motor pathology, such as, for example, Parkinson's disease. We know that in the substantia nigra, you find a lot of dopaminergic neurons, in particular in the nigrosomes, in particular in nigrosome 1, which is basically like an area where you have high density of these neurons. And we also understand that you have a high loss of dopaminergic neurons in nigrosome 1, coming with Parkinson's disease and starting a long time before the actual symptoms arise. And we were kind of motivated by the fact that we already know from other studies that there are clear signs for Parkinson's disease that can be measured with MRI here, for example, T2 star weighted contrast, the so-called swallowtail sign, which you see in healthy controls, this hyperintense area, but that disappears in Parkinson's disease. But to really fully appreciate what you have here, what we need to understand is the precise mechanisms. Why does the contrast change? What does it what drives the contrast? And can we actually model it quantitatively? And that's what Malte Bramelow set out to do in our department. And he first started off with a postmortem experiment and did histology postmortem and very high resolution MRI in collaboration with Jürgen Reichenbach's lab in Jena. So we actually have 22 micrometer resolution MRI data here. And it really looks like the histological data under the microscope. It's quite amazing in my opinion. And why do you see, what are these dots? These are the dopaminergic cells. They're filled with neuromelanin pigments that carry a lot of iron. And that's why you see them both under the microscope because of the pigment, but also in the MRI because it contains a lot of iron, which drives the contrast. And you can see why it drives the contrast, because it distorts the frequency locally, and MRI is sensitive to that. So this was really encouraging that we can see that directly. But the problem is obviously in vivo, we are not going to acquire 22 micrometer resolution data. So what can we do? We can try to model this again using the appropriate biophysical models. And I can only gloss over this here now in the presentation, but please feel free to go to the pre-recorded talk where I go into more details. But fundamentally what Malte did, he did, again, performed quantitative histology at where he recorded data of iron concentration at cellular resolution, at 1 micrometer resolution. And he used that to calculate and predict the MRI signal using the appropriate biophysical models. And you can see how well the actual predictions fit the MRI data he also recorded in the same samples. And what is now important to realize is that with that static defacing model he implemented, you have a direct connection between the measurement here in MRI and the iron concentration in the dopaminergic neurons. And you can exploit that. And here's an example, or maybe another way how to show this. When you look at R2 star contrast and R2 in the nigrosome 1, you can see that it's dominated when you take the difference between the two, which is called the R2 prime, then that difference is dominated here by this microscopic component of the neuromelanin contrast, iron-driven contrast. And the neuromelanin is primarily in such high concentrations in the dopaminergic neurons. So now you have effectively a marker, direct relationship to the iron concentration in the dopaminergic neurons. And this could potentially be really an interesting biomarker in terms of iron accumulation in Parkinson's disease in those neurons. But if it's calibrated in the right way, maybe even something like a cell density marker of dopaminergic neurons. So a marker for neuronal loss in Parkinson's disease, early biomarker. But to get there, obviously, it's not enough to just build the generative model, even though it's quantitative, highly validated post-mortem, but that doesn't suffice to bring it into in vivo. To get it into the in vivo situation, you have to overcome a few more quite significant problems, as we all know. And one of them is, for example, to identify these small areas, the nigrosomes, reliably in the substantia nigra, so we can detect them. We have to push the resolution. We have to check the models in vivo as well. But I think it's worth it. It's a really interesting avenue. And if you want to learn more about the substantia nigra and kind of identifying it precisely, look at Malte Bramelo's poster, please. So let me wrap up my presentation here with a brief discussion. So I hope I could convince you, even with the short form of the talk, that MRI can characterize microstructure features, especially when we have developed the right biophysical models for that. And this is a non-trivial task. When we have that at our disposal, we can run these longitudinal large cohort studies of microstructure. And indeed, a lot of studies have already been performed in this area. I just didn't have the time and opportunity to present those, but please look at the literature and also at some of the presentations here at OHBM. This enables us to look at microstructure function relationships, which excites me a lot because we can really look at the entire brain potentially here. We may have early biomarkers of neurodegeneration, especially with the right biophysical models. It's a versatile and transferable tool. We can use it in vivo, post-mortem, apply it to different patient and subject groups. We can look at different species. But to really push it and make it a systematic in vivo histology approach, we still have to overcome some major challenges. One is further pushing the quality of MRI, of resolution, also dealing with the large data sets we have. So the raw data for some of the data I presented is about 1 to 200 gigabytes in size. So we have to deal with this properly. We also need the right histological reference data for cross-calibration, for validation, for referencing, and we're really lacking this. We need more quantitative and three-dimensional data here. We also want to do validation in vivo, because obviously the post-mortem situation, the in vivo validation are quite different, and they could also mean that the contrast mechanisms are actually different. So we need this as well. And I presented some examples in terms of looking at structure-function relationships that could help with this type of validation. A big challenge is unifying the biophysical models across contrasts. So ideally, we'd like to have one grand unified model that explains all contrasts based on all types of histological microstructural features. And then conceptually, a problem that does not only relate to us as a, let's say, MRI community, neuroimaging community, but in general, the neuroscience community, we have to deal with these multiple orders of magnitude of spatial scale to understand the brain. But if you just wanted to take away maybe one message from this talk, then I would say it's the one that I think it's really exciting times, because I think we are now pushing MRI from, let's say, a qualitative imaging device to a quantitative measurement device of physical parameters, kind of a scientific measurement instrument, and even further beyond that to a measurement device of biological characteristics. And this is, of course, a major endeavor, not only performed by us, but by a large community of researchers, and would be impossible without good and well-working collaborations, highly interdisciplinary collaboration. So I would really like to thank, first of all, the members of the Department of Neurophysics in Leipzig, and then also the many collaborators we have that have helped us with a lot of these experiments and big challenges we face. And I would like to thank you for your attention, and I'm looking forward to the questions.
Thank you very much, Nick, for this very clear and thorough presentations. And it's quite a feat that you can present advanced quantitative MRI and it looks as easy as walking in the park. in a way.
But it isn't, as we all know.
Yeah, that's the hard wall of reality means that those results come from weeks and efforts of many people to achieve these. Yeah, I see that there have been a few questions in the chat, and two of them I'll read them out loud, and then maybe I'll rephrase them. So there is one from Hiromasa Takemura. He wrote, I think that obtaining quantitative maps using 7T has some challenges, such as large B1 plus inhomogeneity compared with 3T. May I ask you to give us a ballpark figure on how you resolve technical challenges in 7T QMRI? The other one is related to that. So can this tech be performed on a 3T machine, or the SNR will be too weak? And overall, my way of wrapping up and launching the discussion would be, what are the hardware requirements to do this? Try this at home is very nice, but what do you need to do it? And at the same time, what can we expect from what we have at home? Which is the obvious question of the 3T versus 7T. You mentioned the connectome scanner, which are not really the most common machines around the world. And yeah, that would be the first point maybe to discuss.
Absolutely. Yeah, indeed, there are only 4 systems installed at this point of the connectome scanner. So it's a relatively unusual system. These are brilliant questions. Let me try to walk through the different aspects. So maybe let me start with the technical problems working at 7 Tesla. So absolutely important point. And I really glossed over that, but there's a lot of work happening in the background to make this work. And one of them is dealing with the biases you encounter at 7 Tesla, but also at 3 Tesla. You also need to deal with them. At 7 Tesla, that's just a bit more, let's say, pronounced than at 3 Tesla. And one of them is, for example, so-called inhomogeneities in the RF transmit field, or the B1 plus field, how it's called. And you can't avoid them because they're intrinsically linked to the fact that you put a brain or a person into the scanner. So what we typically do with, and there are other examples for these biases, but what the typical approach with quantitative MRI is that you do not only acquire your basic imaging data. Let's say here, for multi-parameter mapping, we would acquire so-called flash or spot gradient echo data, but we acquire additionally reference data, something like, for example, RF transmit field maps, B1 plus maps, or also receive field maps, to calibrate that, to kind of let this enter into the calculation. And there are now quite a few tools out there, open source tools, that allow you to, when you have these additional reference data and your imaging data, to put it together and calculate these corrected maps from it. Like, for example, the HMI toolbox would be an example you might want to look at. So, but this is crucial to do this. And even having done this at 7 Tesla, you still hit the limit at some point. I mean, there's still areas that are really hard to image, like deep areas in the temporal lobes would be an example for that. Or there are residual effects, and now we are really getting into the nitty-gritty details, but it's important to realize that a lot of these contrasts we measure, they keep interacting. We try to really quantify them and separate them, but they interact at the level of the biological tissue, so to say, in the compartments, and it's quite hard to tease them out. And one effect is, for example, magnetization transfer effects at 7T that can be more pronounced at 3T. And are sometimes undesired. Sometimes we want to image them, sometimes we don't. So there are all these details, but I think we are making really good progress as a community. And maybe the last point regarding technical problems, I think I put a great deal of hope and promise into parallel transmit techniques, for example, dealing with B1 inhomogeneities at 7 Tesla. They're very useful for that. Now, coming to the question, does this work on more, let's say, typical MRI devices, so going to 3T or lower gradient amplitudes? And some of these techniques, indeed, they do. For example, the cortical myelin mapping example I showed initially, was acquired on a 3T KIM Trio system at the time, so relatively standard MRI scanner. And that works. What is difficult is the resolution is limited. So that was data acquired at 800 micrometer resolution. So if you want to look at, for example, the layer effects, you'll be rather limited. But if you look at average, so to say, myelination in the cortex, this is a pretty good measurement. And sometimes it's really non-trivial to tell whether the 7T data will actually be better than the 3T data, because these contrast mechanisms, they depend in a non-trivial way on signal to noise, and both is manipulated when you go from 7T to 3T. So sometimes you can even have an easier job at 3T. So don't give up on that. Have a look at the literature, but definitely these kind of myelin maps, putting them on the cortical inflated cortex, they work rather nicely at 3T, I would say.
Yep, that's very compelling. And yeah, there are limitations, and with the no large access to 7T scans, that's probably where further development could happen, I believe. And in terms also of antennas and hardware, I mean, there's been progress at 3T, but then at 7T, it's still quite a new field. Are there some technical things that you wish were existing for your 7T systems that are not available yet?
I think what would be really beautiful, and I think there are interesting developments, for example, when you look at David Feinberg's work in California on that, is what would be nice is to kind of bring together the capabilities we have on the connectome scanner 3 tester with a high gradient strength and have that available at 7 tester. So something like a 7T connectome MRI scanner. And I think there are really interesting developments in terms of head gradient calls and so on. So maybe not really so far away anymore. So that would be nice. And I think there have been great developments in terms of making PTX, the parallel transmit methods, more readily available, more easy to use, kind of bringing it into a production system. And for us, this is really an important aspect. And I didn't focus on this, but I'd like to make this point. It's not only about developing all these super fancy techniques and applying them then maybe in a couple of subjects then in the end, or participants, or patients, but we really would like to create techniques that can be rolled out more widely and used regularly, and thus usability is an important aspect as well.
Thanks. Coming from the audience, lots of very positive comments. I think everyone really enjoyed the talk, but that we knew that when we invited you.
Flattering too much.
There are still some interesting scientific questions. Again, I'll try to wrap up 2 questions from Sridhar Narayanan and Andrew Alfie Wern. I'm trying to catch up with all the questions. So there was a question from Sridhar about, do you obtain R1 map via MP2 range, since different R1 mapping approaches yield different results? Does the accuracy of T1 or R1 mapping matter, or is internal consistency sufficient? And I would link that with Alfie, who's asking a similar question, but in terms of T1, R2, myelin, and so on. But in case of pathology, where many changes are occurring simultaneously, cell death, edema, and so on. So do you think these quantitative maps are still both sensitive and specific to these singular elements of microstructure? So that's a question about those parameters. We measure the physics, but how do they relate to the underlying biology in healthy subjects? In a way, how reproducible is that? And if you change anything in the sequence, do we get something completely different? And what is it worth in terms of lesion tissue or pathological tissues?
They are great questions. Also really difficult to answer, I must say. But maybe let me start this way. I think it's absolutely correct that currently, when you use different T1 mapping techniques, you might not get the same results out. They're pretty similar, and also the features and so on really nicely comparable, but the numbers you get out, they would differ. This is not unsurprising. And there are several reasons. One of them is technical. There was a question about MP2 range and so on. So some of them are technical aspects, but I think the more fundamental one, and that makes it hard, even harder, is it goes all the way down also to understanding how the quantitative physical parameters link to the tissue. Because we are basically, even though we call it R1 measurements, they're all done slightly differently, and they slightly differently probe the tissue. And that's part of the reason why you get different R1 values out. And this could be, again, coming back to this point of, this could be magnetization transfer effects that come out differently in these techniques. And so this goes really back to this biophysical modeling understanding, how the data are generated, basically. And I think the better we understand that, the better we'll also do in terms of the quantitative MRI imaging. And I think this brings me straight to this question about the pathologies now, and because it is about the biophysical modeling. And I think the same is true here. I'm probably less worried about the sensitivity of MRI than the specificity. I think we're actually quite sensitive to a lot of things that are happening, but then we often encounter this issue. What does it mean? How do we interpret it? That's the big issue typically for us. But I think there we also really have, in principle, we have an approach towards that. And I would like to refer back to, for example, to the example of the substantia nigra and developing a model for the substantia nigra and the dopaminergic, the ion concentration there. There you could see how you can set it up by using the right histological data, studying your tissue and your, for example, particular brain area very precisely, understanding there the contrast mechanisms. That makes it much more specific what you get out. And you also understand maybe when it fails and it's not specific. And in an extension of that, and I think that's important, and that's something we are working on now for the substantia nigra, you also want to understand this as well as possible in the pathological cases. So what happens there? And it's absolutely right. I mean, a lot of things are happening typically in the pathological tissue that might interact, that might cancel each other out. But I think I'm still quite optimistic with the right biophysical models and using a multi-contrast approach that is sensitive to the different aspects. I think we can tease out quite a lot of information there.
Yep, thank you for that very clear and detailed answer. So in a way, do we need to get more MR biologists more than MR physicists? That would be the next trend, or neuropatholo MR experts in order to bridge the gap between that, because that's the thing. You've got the super nice but really deep MR physics, and on the other side you are looking at something that are really happening at the level of the cells, at the core, the biology of the cells. And those two fields are not taught together in a way. So
I agree. I think this is, in terms of the training and teaching aspects, quite challenging, because when you look at the physics, I mean, the kind of questions you try to answer, they're not so dissimilar to what a lot of people in, let's say, basic fundamental physics do in the areas of theoretical physics. You know, these aggregate measures, coarse graining models, they're not so unusual in that field. But what is highly unusual is to have people who work in theoretical physics coming all the way over into this domain of biology, for example, and vice versa. So, so I think in terms of the training, we're probably looking at an even bigger challenge in terms of interdisciplinarity and pushing that further. And this is one reason why we have such a big host also of collaborators, for example. And we couldn't do this work by ourselves. Because it's so interdisciplinary.
Yeah, there is so many things that still need to be discovered and done. That's good for the younger people incoming in the field.
Exciting times.
Yeah, well, I think it has been exciting for the last 20 years, for me at least. So I hope it goes on for another 20 years when I retire. That will be perfect.
That would be great, yeah.
More questions. Two questions again related, a bit more of the ex vivo imaging. That's from Andrew Reid. Fantastic stuff. Have you compared the high-res diffusion tractography with polarized light imaging data? And I think there was another question. That was from Sridhar. Could you comment on the impact of fixation on postmortem qMRI and what it means for translation back to in vivo imaging? So yeah, so to validate, we extract the brain, but we need to fixate it in a way. So how does that affect things? And I know that PLI is also a exciting technique because it goes even with higher resolutions, and latest developments let you look inside the brain even with more details, but they're all ex vivo, which imposes those extra constraints. So how does that match what we do in vivo?
Mm-hmm. Mm-hmm. So let me start with a polarized light question. So we haven't done this, the direct comparisons. Other groups have started looking into this. For example, in Oxford, Carla Miller's group looked into that. And also, of course, in Jülich, they do a lot of work on the PLI, on the big brain, and I think very exciting work. And indeed, we were performing some of the scans I presented here, some of the approaches on one of the upcoming big brains, so to say, so the next generation. So we hope that we can bring this together more closely, these quantitative MRI techniques with these really advanced whole brain histological techniques, for example. So we haven't looked at PLI, but we started looking at, for example, tracer studies. So we are quite interested, for example, validating the U-fiber measurements I've shown post-mortem using tracers, for example. So we started to do that. But obviously, this can only be done then post-mortem. So you have this kind of limitation of what happens then post-mortem and with fixation. And so the fixation effects, they're obviously significant. They have a significant impact on the signal you measure. That looks very different from the in vivo situation. So, for example, the relaxation times they often shortened, and you also have effects on the diffusivity because of these cross-linking effects of formalin. I think the really difficult question to answer, and that's why I said we really need to do these in vivo experiments on top of it, is not only whether the parameters change. That wouldn't be so bad. That just means the parameter value has changed, but the contrast model is the same. But what is the worry is that the contrast model itself might change. And for example, that you might see certain tissue compartments, you might see them in vivo, but not post-mortem anymore. And that means you have to then adjust the models. And I think that's more critical. The other thing which can be rather awkward is, for example, that fixation typically doesn't happen immediately across the entire brain, at least when you look at human whole brain post-mortem scans. And so you might have differential developments of the fixation, depending on where you are in the brain. So you get biases as well. And for example, Siyavush Mohammadi's group in Hamburg looks at that. So I hope
a
bit of an idea.
Yeah, perfect. I think we're reaching really the end of our hour. And there was, well, obviously, looks like we've been cut off in the main room, but then I think this is recorded, so we can still wrap up very briefly. There was a question again from Sridhar about potential downside to prospective motion correction in the context of qMRI, and as a wrap up question, I would like then to expand to, okay, if we scan well-behaved subjects, we can expect they don't move. But when we translate from MR physics to research to clinical, we'll have to deal with not so friendly or not friendly, not so well-behaved subjects. So movement will be an issue, and this PMC could become necessary. And how do you see that? And with that, we can wrap up things. You've got 1 minute, pretty much.
Okay, I'll try my best. So how I would see it is, I think if you have really well-behaved and you know, kind of highly trained subjects, you probably get away with not using something like prospective motion correction. But even then, you will hit limits because it's really hard not to move. I mean, we talk about voxel sizes of 500 micrometer. So it's really tiny voxels, and even breathing and the heartbeat will move your head a little bit. So I think motion correction is key. We need to really have good motion correction. In terms of the patient studies, I fully agree. The reality is, I think there you really need some reliable motion correction, be it, you know, optical prospective motion correction, maybe some post-processing technique, image reconstruction, a lot of techniques out there now, but we really need that. And what we've learned from studies on dementia patients is that one should keep this in mind. Motion does not equal motion. It depends on your patient population, perhaps on your scanner, the type of scans, how subjects and patients move. And one of the motion correction techniques might work really well on one particular group, but pretty poorly on the other one. So it becomes also a higher dimensional and more complex problem, and you really need to tailor the technique. I think that's important to keep in mind.
Thank you very much. I think we've just hit the gong now. So Nick, it was a pleasure to share the screen here. You did a fantastic job, and I hope to catch up sometime in real and in Leipzig or Liège or wherever around the world. Thank you.
That would be really nice. Yeah. Thanks. It was an absolutely pleasure and great questions. Thanks. It was a great discussion. Thank you, Chris.
Thank you. Yeah. Thank you. Cheers.
Thanks. Bye.